There are a variety of species in our environment, including large, small, and microscopic ones. As humans, we manage to coexist with them and must have some defense mechanism to keep ourselves safe. Like how humans evolved, they built houses to establish a barrier between themselves and the harsh environment. The body has its own defense system that fends off any external pathogens. It is something that the species are born with. This defense system of the body is known as the immune system, and the ability of the immune system of protecting the body against infectious disease is known as immunity. Immunity comes in 4 different forms. Active immunity is also known as immunization, passive immunity, adaptive or acquired immunity, and innate immunity. The immunological response you have from birth is referred to as innate immunity. This means that activation is not necessary for such a response. The innate response is quicker. Adaptive immune responses require time to develop and are only developed after exposure to a particular pathogen. When a person receives antibodies against a disease, rather than developing them naturally through their immune system, this is known as passive immunity. Through vaccination, active immunity is developed. The question that we will answer today is, what is innate immunity? It's not a simple question that can be answered in a few lines. But I will help you understand the mechanism of innate immunity by walking you through the components that make up the innate immune system. Let's dive right in and discover the answer to our query. The word innate means inborn and natural. So, the term innate immunity is the kind of immunological response that has been present since birth, as the term would imply. We can describe it as a quick reaction that does not need to be activated, unlike the adaptive response that needs time to carry out a response against a pathogen. To understand it more easily and look at its components, we can categorize it as defense against entry into the body, and the defense once the microorganism once it penetrates the body. Additionally, it goes by the names first-line defense and second-line defense. As a result, the body's first line of defense, or defense against invasion by pathogens, is composed of basic physical and chemical barriers. Let's think of it this way. In order to enter the body, an infectious agent must get past physical barriers and biochemical that are present at the surface of the body. If we look at the physical barriers first, the skin, which is often impenetrable to the majority of infectious pathogens, is one of the most significant of them. The direct inhibitory effects of lactic acid and fatty acids contained in sweat and sebaceous secretion, which lowers the pH, and prevent many bacteria from surviving for an extended period of time on the skin. We might say that the initial defense against any alien virus is the skin. However, infection becomes a significant issue when there is a loss of skin integrity, such as in cases of open wounds or burned skin. Secondly, we have the mucous membranes provide a mechanical barrier to pathogens and other particles at body openings. Mucus, a protective barrier that prevents pathogens from adhering to the epithelial cells and entering the body, is secreted by the membranes lining the inner surfaces of the body. Mechanical methods like ciliary movement, coughing, or sneezing can be used to get rid of the microbiological infections trapped in this mucus. Those were the physical barriers that play their part in innate immunity. Now, if we look at the biochemical barriers, a lot of the bodily fluids that are released contain microorganisms or antimicrobials. As for the antimicrobials, they are short-chain fatty acids and peptides act as antimicrobials. White blood cells and epithelial cells produce the short chains and peptides of these fatty acids. One example of antimicrobials is the lysozyme. They are found in tears and nasal secretions, as well as acid in gastric juice. Another chemical barrier is the production of sweat and other acidic substances that further strengthen the defense of the skin barrier. A very thin, slightly acidic film is created on the skin's surface by the acids secreted by sebaceous glands in the dermis of the skin. As mentioned before, it can be considered a chemical barrier. This layer acts as a barrier to stop bacteria, viruses, and other hazardous substances from entering the skin. Urine and vaginal secretions are also too acidic for many pathogens to endure. Last but not least, the body's normal flora contributes to defense against infection infiltration. The natural flora's organisms defend themselves against outside infections in 3 different methods. By taking up space, they prevent a variety of potentially hazardous diseases from growing at superficial places. They either compete for nutrients, release inhibitory chemicals like acids, or both. That ends our discussion on the biological and chemical barriers that serve as first line of defense in the innate immunity. So, does this mean we have answered our initial question of what is innate immunity? No, because just like I mentioned before, it's not a simple question. Of course, now that you know about the existence of first and second line of defense, a new question comes to mind. What happens if the first line of defense fails? And what is the second line of defense? In case the first line of defense fails, such as after a cut, and integrity of skin barrier is compromised, then microbes and potential pathogens can enter the body and can begin to multiply rapidly in the warm, nutrient-rich systems, tissues, and organs. That is when the second line of defense comes into action. To get rid of invaders, the innate system rapidly initiates 3 types of non-specific mechanisms to fight them off. The innate system employs non-specific cells like phagocytes that perform phagocytosis and chemicals like complement-related compounds. Another defense mechanism used by the body to repel an invader is inflammation. In summary, phagocytosis, complement activation, and inflammation are 3 ways that the innate system carries out its task. Let's examine these 3 in more detail. You must have heard about immune system cells, or cells of immunity, in your studies of the immune system. But so far, I have not mentioned any cellular fighter in the first line of defense. The cells of the immune system are a part of the second line of defense. Non-specific phagocytic cells are alerted whenever the first line of defense is breached and the pathogen enters the internal environment. These cells can recognize pathogens due to the presence of receptors. These receptors are known as pattern recognition receptors because they can recognize and bind to the surfaces of microorganisms that typically bear repeating patterns of molecular structure. The repeating pattern is also known as pathogen-associated molecular patterns, PAMPs. The phagocytic white blood cells, or leukocytes, such as macrophages, which congregate within minutes, start to attack the invading foreign microbes. They kill the invading pathogen through a mechanism known as phagocytosis. Now if we break down the word phagocytosis. Phago means to eat. Cyto means cell or hollow vessel, and osis means condition or process. Thus, phagocytosis can be defined as cell eating. To put it another way, it is the process through which phagocytic cells ingest the invading bacteria. The process of phagocytosis takes place in steps, but at a rapid speed. There are 3 steps in the process of phagocytosis. The first step is the attachment of the phagocytic cell to the microbe. Sensing the chemotactic signals in the form of chemokines and cytokines, phagocytes like neutrophils reach the site of infection or injury. Once they arrived at the infection site, phagocytes are now very close to the microorganism. Here the process of phagocytosis activates, and this activation is triggered by binding through one of the receptors present on its surface, known as pattern recognition receptors, PRs. In general, pattern recognition receptors on phagocytes can detect pathogen-associated molecular patterns, PAMPs. These are actually a diverse set of microbial molecules that share a number of different general patterns, or structures, that alert immune cells to destroy intruding pathogens. Normally, the most common PAMPs are usually mannose-binding lectin, MBL, which are the complex carbohydrates found in the cell wall of yeast, and lipopolysaccharides, LPS, which is the cell wall component of gram-negative bacteria. This mechanism of attachment between PRs and PAAMBPs is common for non-capsulated pathogens. Following attachment, the process of phagocytosis moves on to endocytosis or ingestion. So, endocytosis is the process by which cells take in substances from outside of the cell by engulfing them in a vesicle. When the foreign particle or pathogen gets attached to the receptors of phagocytes, it is then pinched inside the cell, together with a part of the phagocytic cell membrane. This vacuole is composed of a phagocytic cell membrane with a captured pathogen inside, and it is now called phagosome. After the phagosome formation, it fuses with one of the characteristic vacuoles of the phagocytic cells. These vacuoles contain lysosomes. The fusion of phagosome and lysosome forms another vacuole called the phagolysosome. This phagolysosome is a microbicidal vacuole, and it contains many substances that have the ability to kill microbes. Within these microbicidal vacuoles, invading particles are further processed. Now we will talk about the third and the last step of phagocytosis, which is the killing or digestion of microbes. After the formation of the phagolysosome, it can break down the microbe inside of itself by different mechanisms. Firstly, it drastically lowers the pH of its internal environment. The phagolysosome's environment becomes extremely acidic when the pH is lowered. In order to prevent whatever is inside the phagolysosome from infecting additional cells, this is an efficient technique to eliminate or neutralize it. It can also neutralize the microbes through hydrolytic and digestive enzymes. The phagolysosome contains a number of enzymes such as proteases, lipases, and lysozymes. They act on trapped microbe and hydrolyze the microbial proteins, lipids, and the cell wall peptidoglycan. The third mechanism of digestion involves the scavenger molecules. Some examples of these scavenger molecules are lactoferrin, sequesters the iron required by some bacteria. Ascorbate, interacts with copper ions and hydrogen peroxide and can be bactericidal. Along with that, this intracellular killing of many bacteria requires the uptake of oxygen by the phagocytic cell. So all these events initiate respiratory and oxidative bursts. So, all these things like acidic medium or low pH, digestive enzymes, and oxidative burst to cause the killing or digestion of trapped microbe, leaving behind a sac filled with indigestible material called a residual body. The residual body then travels toward the boundary of the cell and is eventually discharged from the cell by a process called exocytosis. After this, phagocytes can also undergo partial digestion of microbial protein, which can be displayed on the surface of the antigen-presenting cell. Phagocytes present the fragments on their surface via class II major histocompatibility complex, MHC class II molecules. Circulating helper T cells recognize these bacterial fragments and begin to produce proteins called cytokines. That's how macrophages play a dual role as phagocyte in innate immunity and as antigen-presenting cells in adaptive immunity. So that was just one defense mechanism of the innate immune system once the pathogen breaches the first line of defense. Another mechanism is the activation of complement proteins. The complement proteins, or the complement system, is a part of the innate immune response. But we see that the complement system acts as a bridge between the innate and acquired immune response, as one of the pathways of activation is antibody-dependent. The complement system is composed of about more than 20 different proteins that work together to destroy invaders and to signal other immune system players that the attack is on. It is called a complement because it complements the ability of phagocytic cells and antibodies. The complement system's primary purpose is infected cell lysis and immune clearance. The system also works to initiate inflammation so that additional immune cells may join the fight against a pathogen and the opsonization of pathogens can take place. Well, the complement proteins assist in pathogen killing via three pathways: the classical complement pathway, the alternative complement pathway, the lectin pathway. The classical pathway is antibody-dependent and thus part of the acquired immune response. Lectin and alternative pathways are part of the innate system, as they can be activated upon first exposure to a pathogen. Well, all three pathways follow the same pattern once the C3 protein is activated. In other words, the pathways differ from one another in the way of their initial activation step, while the rest of the pathway is the same. The protein fragments that are formed when the complement cascade is activated serve a variety of purposes, including enhancing inflammation and the opsonization process. So, here we are just going to skim through two of them that are part of the innate immune response for revision. Starting off with the alternative complement pathway. After spontaneous activation of the C3 protein, it forms a complex with other complement proteins. This complex can directly bind to bacteria. Once bound, C3 initiates a cascade of cleavage. It forms C5 convertase and C3 convertase. Further cleavage of complement proteins results from forming of complement complexes, which ends in the binding of several complement proteins to the surface of the bacterium in the form of a membrane attack complex, or MAC, or can generate opsonin that labels a bacterium for destruction. Then, a membrane attack complex, or MAC, can insert into the cell membrane of pathogens. There, it produces pores that allow the entry of membrane-damaging molecules, such as lysozyme, and makes the bacterium susceptible to osmotic lysis. Now, moving on to the lectin pathway. During the lectin pathway, mannan-binding lectin, or MBL, binds to proteins containing mannose residues that are found in some types of bacteria, such as Salmonella species. Once bound, MBL forms a complex with an enzyme called MBL-activated serine protease, or MASP. In this form, this enzyme activates C3 convertase, which participates in forming a membrane attack complex. And the rest of the pathway is the same. In our video on the complement system, we previously had a thorough overview of these three pathways. If you wish to go through these pathways step by step, check it out here at sqadia.com. Finally, We will now talk about the last mechanism through which the innate immune system forms the second line of defense. Yes, we will now talk about inflammation. Inflammation is one of the immunological responses produced by the immune system in reaction to an invader. Let's look at its definition. Inflammation is a non-specific host defense mechanism that combats infection by all the means at its disposal. Other than infection, it also responds to any type of tissue damage or injury and resolves or repairs it. The type of white cells involved in the inflammatory response are neutrophils, macrophages, eosinophils, or types of lymphocytes, like T or B lymphocytes, vary with each type of inflammation. So, we can say, inflammation is a normal response of the body to protect tissues from infection, injury, or disease. Mechanism of acute inflammation, which supports the innate immune response, is dependent on inflammatory mediators, also known as cytokines. Remember I mentioned them before. When pathogens breach the external protective barriers, the initial reaction to those pathogens is usually an acute inflammatory response, involving an influx of leukocytes, complement, and other plasma proteins into a site of infection or injury. And these influxes are triggered by inflammatory mediators. Here, a complex variety of mediators are involved in acute inflammatory responses. After release of inflammatory mediators, the leukocytes that are charging along in the blood vessels need to be redirected to migrate into the inflamed site. So, once leukocytes are migrated towards the site of inflammation, then there starts the initiation of the acute inflammatory response. Locally, the acute inflammatory response include vasodilation, exudation of plasma proteins that leads to edema, leukocyte adherence to endothelium by the help of various chemicals and receptors, chemotraction and local activation of leukocytes. Other than the acute inflammatory response, the process of inflammation also includes the acute phase response. In this, various cytokines are produced that carry out signals to other cells of the immune system and also raise the temperature of the body, which is unfavorable for the invading pathogen. So, this was an overview of all inflammatory responses. And with this, our answer to the question, what is innate immunity, ends here. You don't need to take separate notes for your later review, because you can easily obtain the PDF version of this lecture at sqadia.com.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.